lecture, I will discuss with you some data we have obtained in, I would say, in the last 5 years. It will focus on how to design and discover a new molecule which can act as MDM2 p53 protein-protein interaction inhibitors. So first, just some statistics about cancer. Cancer is second disease overall the world. In the report into 2020, it has been reported that around 18 million new cancer incidences and 9.6 million died in 2018. So it's really, I would say, horrible disease. So it's a challenge to discover and develop a new molecule can therapy this disease. So our research focused on, or started in 2014, to design a new molecule which can react as MDM2 p53 inhibitors. So once the cell has been stressed and damaged, so some changes take places in DNA, then the cell become cancers. So what will happen after the cell become cancer cells? Automatically in the body, there is two proteins called MDM2 p53. Once this cancer cell occurred, automatically p53 generated, then we have possible two routes, either cell cycle arrest and DNA repair, then cell cycle restart and become a normal cell. The second way is just to kill these cancer cells, then eliminate from the body. This mechanism make the cellular and genetic stability of the body. So once the cancer cell started, so as I said, we have two routes. The first route either repair the cell, and this via signaling some signals to give some orders to repair the defect which it has been affected in the cell, or make another signals to kill these cancer cells and make the genetic are stable. So by X-ray crystal structure shown that the connection between MDM2 and p53, there are some amino acids were found in the binding between these two proteins called phe19, tryptophan 23, and leucine 26. So this is a hydrophobic pocket between these two proteins. So to design a molecule to disconnect this connection between these two proteins and liberate or dissociate the p53 to do the job for tumor suppressor, this is really a challenge. In the reported literature, there are some scaffolds have been reported and shown potential activity towards this protein-protein inhibitors. As you can see here, Nuclein reported by Roche, and others by research groups. And if we can look at this structure, we can notice that there are some moieties are essential for binding in the pocket, like aliphatic group or some halogens and like that. So our research group has been focused in this area, and the most candidate and promising to do this job is Spirooxindole scaffolds. And one of the most efficient approach to synthesize this Spirooxindole is 1,3-dipolar cycloaddition reaction. This efficient method and convenient in just one-step reaction mix 3-component reaction to access the final compound. The final compound has contiguous stereogenic centers, and the biological activity of this final compound is really promising. The first example I will show you in this area, we have started with MS students, and by search we have found that this molecule is not reported so far. So the final compound is quite easy to prepare. If you can start from acetone with aldehyde, we can make it chalcone very easy. Then the second step is just to mix the chalcone with thioproline and isatine, heating 1 to 2 hours. We can access to the final compounds in 89%. The structure has been confirmed by spectrophotometric tools, including the X-ray crystal structure. Then the mechanism to form this final compound is the first step is just reaction between secondary amine and the most active carbonyl in the isatine ring to form intermediate by heating, losing carbon dioxide to generate the azomethane ylide. This is the active species which can react with the chalcone either two ways to provide the final compound in radioselective and distereoselective fashion. Then we have scoped the substrate with different substituent of aldehyde. We have prepared around 16 compounds to study the structure activity relationship. Then we moved further to examine this compound against some of cancer cells, including colon cancer, liver cancer, and prostate cancer cells. And here in this table, the results we have obtained, and we have found that the hybrid 4D is the most active compound between this series, and shown that 1.4 micromolar compared to cisplatin and selectivity index with 3.6. For the second cancer cell, liver cancer cell, also the same compound shown the most active compound between these series. In the third cancer cell is a prostate cancers. We have found that 4N is the most active compound compared to this cisplatin. Structure activity relationship between these compounds, we have noticed that a halogen on the phenyl ring make a significant change in the activity, and also nitro group give a better activity in this cancer cells, colon cancer cell. The same halogen on the benzene ring make really significant effect on the activity. For example, this compound with bromo and fluoro and 2-chloro on the position 2 and 4 on the benzene ring. The same for prostate cancer cell, also trifluoromethyl group, it was the most active member of this compound. Then we moved further. For the most active compounds, we have done a set of bioassays, including colony formation assay, cell cycle arrest, and wound healing assay, anti-apoptotic BACs and Bacil-2 proteins, caspases, and cell cycle inhibitor for the p51 gene. To understand the mechanism of this compound, how it works. So this figure shows the IC50 for the most active compound for D for 3 different cell lines. Here, this is the colony formation assay against colon cancer. And as you can see on the left, this is a colony. And after using 2 micromolar of this compound, it can kill all cancer cells. Further wound healing assay, as you can see here in this figure, figure C, after treat the cancer cells, after healed the media, with 1 micromolar for 20 hours, no growth of the cancer cells. Then we have carried out cell cycle arrest, and as you can see here in this figure, that the most active compounds for D with 2 micromolar of compound with 24 hours can arrest the colon cancer with 34.5 Ng2M phase. To be sure that the p53 level has been generated, we moved further and make this assay. And as you can see here, with 1 micromolar of this compound and also with 2 micromolar of this compound, and has been shown that a level of p53 have been increased up to 52.7% and 61% respectively. Moved further, that p53 once generated, it can give another signals to p21, and to confirm this one, we performed this assay, and after 12 hours, we can notice that the percent of p21 gene has been increased. So further experiment to confirm the reaction mechanism of this compound, this BACs and Bacil-2, and from this figure also you can see that increase in BACs and decrease in Bacil-2. Further, to confirm the reaction mechanism, we have been performed the caspase 9, 8, and 3. And as you can see from this figure that the percent has been increased, it means that the compound can induce the apoptosis through mitochondrial and death receptors. And here, the molecular docking of this compound compared to standard drug, and as you can see here that our compounds also can fit with the pocket via hydrophobic-hydrophobic interactions. This work has been patented in USA patent, and later we publish in bioorganic chemistry. In this area also we have carried out the in vivo study of this compound, and we make also nano formulation of this compound, and we have really interesting results which submitted again for US patent as a second patent in this work. Another example I will show you briefly, this another molecule we have synthesized by our research group. Is just mainly we are just playing with this three-component reaction, changing the chalcone moiety, and we have tried to fix or using isotene derivatives and amino acids. The same reaction goes through the same mechanism, and just mix three compounds in one-step reaction, heating. You can access the final compound in very good yield, a clean reaction, in some cases just precipitation and filtration. This series of compounds has been examined against two cancer cell lines, breast cancers and leukemia, and we have found that the hybrid VF is the most active, and VK in both cancer cell lines. This work has been published in Bioorganic and Medicinal Chemistry in 2017. We just switched or used another amino acid, L-proline, and we access to this final compound. We have really nicely X-ray crystal structure to confirm the final compounds. Again, the same. Again, the series have been examined to the same cancer cells, and compound VIJ is the most active against MCF7, and the VII, this is the most active against leukemia through compared to 5-fluorouracil. This work again has been published in photochemistry and photobiology. Another example, as I said, we just trying to change the chalcone. So we have tried to introduce another pharmacophore in the final scaffold of Spirooxindole compound. In this work, we have introduced the indole moiety, and we have prepared series of this compound. This compound has been examined against set of cancer cell lines, and the data has been published in Bioorganic Chemistry and RSC Advance. I will just discuss with you our recent research in this area. This work is not yet published. We just in the final stage to prepare the manuscript for submission. And this is the work for PhD student started almost 2 years ago. This compound has been reported on the left as a potent and stable MDM2 inhibitors. And this on the right, this compound is a natural product and also recognized and discovered as a BCL2. So we are trying to introduce and combine these two scaffolds in one compound, and to see how this compound, biologically activity of this compound, how it will be. So again, we have prepared this chalcone starting from 2-acetyl N-methylpyrrole, again mixed with substituted isotene. We have carried out with different amino acids like pyrrole, thioproline, sarcosine, and saturated indole carboxylic acid, and also so around 5 or 6 amino acids with different isotenes, and again with the same strategy, with the same method route, it can access to the final compound. X-ray crystal structure confirm the final compound. Then we have done several assays, including we submitted this compound to a National Institute of Cancer, and they screen against 60 cell line. then we have found that this compound can be active against 3 cancer cell lines, including breast cancer, liver cancer, and colon cancer. And again, we have carried out some more analysis to confirm the reaction mechanism of this compound against MDM2P53. So I will show you some results. And this is the safety profile of this compound, and also the IC50 of these compounds. We have noticed that these compounds are totally safe, except some, not too much, but 2-3 moieties are not safe, but mostly are safe, as you can see from EC100. And also the activity, it shows like nanomolar scale, and we have found that the hybrid VIIJ, this is the most active compound between this series. I just show you the MST results of these compounds with collaboration with Professor Alexander Domling from Netherlands, one of the most pioneer in this area. He examined our compound and he provided recently the results. And yes, this compound can be react as MDM2P53 inhibitors. And as you can see here in this slides, one on the top left, 1.32 micromolar, on the right down also 1.72 micromolar. Again, this compound with 1.13 micromolar, and we have found that this is the most active compound with 0.612 micromolar compounds. We just compiled this data and it will publish soon. The last example I will show you, and I'm really sorry if I'm a little bit late, we tried to study another example with introducing this benzophenone moiety. Again, we have to prepare the chalcone mixed with azacine derivatives and amino acids. You can access the final compounds, X-ray structure, confirm the structure of these compounds. Again, we have carried out set of biological assays, as you can see here in this slide. Just I will show you that this compound also can be binding with these two protein and generate the P53 to do the job. Here, this is the results of these compounds. And we have found that if we make this compound as salt, and the reactivity really very high, as you can see, 0.0531. Again, some results. So in conclusion, we really have successfully developed a promising candidate based on Spirooxindole, which can work as MDM2P53 inhibitors. We are still working in this area. We are exciting to achieve more than what we have. At the end, I would like to acknowledge King Saud University, which allow me to do this work, especially chemistry department. And also I would like to acknowledge for funding part of this work presented by CAXT, and also people who cooperate with me to do the biological activity, because the facilities of biological activity is not allowed in our research group. So the research group, I would like to thank all people involved in this work, and especially for collaboration with people who've done all biological activity, Alexander Domling, Professor Iqbal Chaudhary, Pakistan, Professor Farid Badria from Egypt, Dr. Fardous from Egypt, and also Dr. Marwa from Egypt, people who helped me to understand the molecular docking, either Prof. Dr. Zahir from Pakistan, and also Yassin and Muhammad Talib from Egypt. The guys who helped us to characterize this compound via X-ray single crystal technique, this Professor Fan, Dr. Hazem Gabbour, and also Dr. Samar and Dr. Fry from Stuttgart, some computational chemistry done by Dr. Saeed, and there's some picture for these guys. And thank you very much for your listening.